Imagine I gave you this, just a line of letters, no blood sample, no microscope, no protein in a tube. Could we actually learn anything biological from it using only a computer? Well, we can start by asking a very simple question. Have we seen anything like this before? And from that one question, this strange line of text can very quickly turn into something much more familiar. A protein, a three-dimensional structure, and eventually something connected to a human disease. So how do we get from this to this? And when do we do that? Are we actually doing biology? Let's start with comparison. A computer can take our sequence and search for similar sequences that scientists have already studied. And biology gives us a good reason to do this. Related proteins often keep parts of their sequence throughout evolution. So if our mystery sequence looks very similar to a known protein, that gives us a useful place to start. In this case, the answer comes back very quickly. This sequence belongs to the globin family. More specifically, it is the beta subunit of human haemoglobin. So we've gone from a string of 147 amino acids to one of the proteins responsible for carrying oxygen in our blood. And we did that without running an experiment ourselves. But there is an important distinction here. The computer did not look at these letters and somehow work out oxygen transport from scratch. It connected our sequence to biological knowledge that already existed. Experiments had already been done, proteins had already been sequenced, structures had already been solved, and all of that information had been collected in databases that a computer can search and connect. That ability to connect biological data is one of the simplest examples of what computation can do for biology. But there is already a problem with the way we're looking at this protein, because a protein isn't really a line of letters, it's a physical molecule. Those letters describe amino acids, but the protein itself is a physical molecule. This is an experimentally determined structure of human haemoglobin. It contains 4 protein chains, 2 alpha and 2 beta subunits. And this beta chain is the molecule represented by the sequence we started with. That gives us another important idea. The same biological object can be represented in very different ways. A protein can be sequenced, it can be a set of 3-dimensional coordinates, it can be a collection of experimental measurements, or it can be represented numerically inside a statistical or machine learning model. None of those representations is the protein itself. There are different ways of storing information about it. And once biology is represented as data, we can start connecting one kind of information to another. For example, we can take one position in the sequence and ask what happens when it changes. One of the most famous examples is a single amino acid substitution in beta-hemoglobin. A glutamate becomes a valine. It is usually called the beta-6 glutoval mutation. If you check the canonical UniProt sequence, you will see it at a position 7, because the first methionine is removed from the mature protein. It is one change in a chain of more than 100 amino acids, but under deoxygenated conditions, that change alters how hemoglobin molecules interact. The mutant hemoglobin molecules can assemble into long fibers, and those molecular changes can affect the shape and behavior of red blood cells. This is part of a molecular basis of sickle-cell disease. So now we have moved across several levels of biology: sequence, structure, molecular interaction, and phenotype. But there is something slightly misleading about this story, because the computer did not discover all of that on its own. The mutation was studied experimentally, the structures were determined experimentally, the databases we searched were built from decades of biochemistry, genetics, structural biology, and medicine. What computation gave us was a way to organize those results, compare them, and connect them. That distinction matters. A computer can make new predictions. It can reveal patterns that are difficult for a person to see directly. But the biological meaning of those results come from their connection to the real system. So a better picture is not computer in, answer out. It is closer to experiment, data, computation, and sometimes that computation produces a prediction that leads to another experiment. We'll come back to that loop, but first we need to zoom out, because proteins are only one small part of computational biology. Take human tissues. Modern experiments can measure the activity of thousands of genes in huge numbers of individual cells. That produces data that is impossible to understand by simply scrolling throughout a spreadsheet. Computational methods help group cells, compare them, and build maps of cell types across tissues. Or take pathogen genomes. If we collect viral sequences from different places and times, we can compare them to study how a pathogen population changes and spreads. And computational biology is not only about analyzing datasets. We can build physical models of molecules and simulate how they move. That is a very different kind of computation. And then there is a machine learning. Models such as AlphaFold can learn patterns from large amounts of biological data and use them to make new predictions. All of these belong under the broad computational biology umbrella. And that is important because computational biology is not just another name for AI in biology. AI is one tool among many. A useful way to map the field is to think about 4 broad types of approach. This is not a strict taxonomy. Real projects mix them all the time. First, there are algorithms and data processing methods. Search a database, compare sequences, align them, assemble pieces of genome, organize millions of records. This is the world we entered when we searched our haemoglobin sequence. Second, there are statistical and probabilistic approaches. Biology is noisy, individuals differ, measurements differ, experiments have uncertainty. Statistics helps us describe that variation and decide whether a pattern is actually meaningful. Third, there are mechanistic or physics-based models. Here we try to represent something about how the underlying system works. Molecular dynamics is one example. We define a physical model and simulate the behavior that follows from it. And fourth, there is a machine learning. Here, a model learns parameters from data so that it can recognize patterns, make predictions, or generate new outputs. The borders are not clean. Statistics and machine learning overlap all the time. Machine learning models can use physical information. Algorithms are needed everywhere. A real project might use all 4. The important point is simpler. Machine learning is part of computational biology. It is not computational biology itself. So, can you do real biology without touching a pipette yourself? Well, yes. You can analyze public experiment data, you can build models, you can simulate molecules, you can develop algorithms, you can make predictions about systems you have never physically handled. Entire research projects can be computational, but that does not mean the computer is disconnected from experiments. Experiments give us observations about biological systems. Those observations become data. We analyze the data, compare it, model it, and make predictions. Maybe a mutation is likely to alter a protein. Maybe 2 proteins interact. Maybe a group of cells behaves differently in disease. Maybe a new protein sequence is worth testing. And those predictions can lead back to experiments. So instead of thinking wet lab versus computer, I think this is a much better picture. Experiment, data, computation, prediction, experiment. Not every project goes through every step, and not every computational prediction can be tested immediately, but biological reality is still what the model is trying to tell us something about. A prediction matters because it refers to something outside the computer. Now, the definition is actually useful. Computational biology is the use of computational methods, models, and biological data to help answer biological questions. That can mean comparing 2 sequences, analyzing millions of cells, simulating a protein, reconstructing evolutionary relationships, predicting a structure, or training a machine learning model. Those methods can look completely different. What connects them is the biological question. This is why I find it more useful to think of computational biology not as one technique, but as a way of doing biology. There is one complication. A lot of what I have just called computational biology is also routinely called bioinformatics. Sequence analysis, bioinformatics, genomic databases, definitely bioinformatics. Protein structures, often bioinformatics. Machine learning on biological data, depending on the context, people might call it bioinformatics, computational biology, both, or something more specific. And there is no perfectly clean boundary that everyone agrees on. The terms overlap and different communities use them differently. So before we go deeper into statistics, algorithms, or machine learning, there is one question we should clear up. What is the difference between bioinformatics and computational biology? That's where we will start next. Thank you.